Everyone, I am Tanmay Reheja from class 10th.
And I am Shobhita Mishra from class 9th.
We both study in Vidya Bharati School, C Block, Surinagar, Ghaziabad, Uttar Pradesh.
We are taking in part ATL Thinking Lab Marathon 2021-22.
Our theme is healthcare and nutrition.
Our topic is innovative solution, specifically solving problems faced by patients suffering from dengue disease in India.
Now, we will talk about the global condition of dengue. Some 2.5 billion people, two-fifths of the world's population, is at risk.
An estimated 50 billion dengue infection occur worldwide annually.
An estimated 5 lakh people with dengue hemorrhagic fever require hospitalization each year.
A very large proportion, approximately 90% of them are children aged less than 5 years, and about 2.5% of those affected die.
Dengue and DHF is prevalent in more than 100 countries in Africa, the Americas, the Eastern Mediterranean, Southeast Asia, and the Western Pacific.
The Southeast Asia and Western Pacific regions are the most seriously affected.
Now, let's talk about condition of dengue in India. It was first observed in Calcutta in 1945 and it became an epidemic since 1963, and it is also observed in almost states of India including Delhi and Lucknow. It is present in almost all states except Himalayan region and Kashmir.
Dengue is a viral disease. It is available in various forms worldwide.
Mosquito vector has a characteristic proboscis and antenna from which it can be differed from other mosquitoes.
See, what is a vector? Vector are living organisms that can transmit pathogens between humans or from animals to humans. They can transmit pathogens from blood.
Dengue is a viral disease and it is transmitted by Aedes aegypti. One develops disease after 5-6 days of mosquito bite. And now let us look at the symptoms of dengue, which are dengue fever, dengue hemorrhagic fever or DHF, and dengue shock syndrome.
Habits of Aedes aegypti are grinding stones, discarded plastic teacups, tender coconut shells, fish tray, flower vase, roof gutters, potted plants, air coolers, earthen pots, water storage cement tank, and discarded tires.
Now, let us look at the vector control measures. Personal prophylactic measures. Use of mosquito repellent creams, liquids, coils, mats, etc. Wearing of full sleeve shirts and full pants with socks. Use of bed nets for sleeping infants and young children during daytime to prevent mosquito bite.
Biological control. Use of larvivorous fishes and ornamental tanks, fountain, etc. Use of biocides bacillus.
Chemical control. Use of chemical larvicides like abate in big breeding containers, aerosol space spray during daytime. We will look at the main methods by which we can prevent dengue.
Environmental management. Reduction and elimination of mosquito breeding source, management of rooftops, porticos, and sunshades.
New tools on the horizon: Wolbachia trans-infected mosquitoes, sterile male mosquitoes, genetically modified mosquitoes, and dengue vaccine. In a nutshell, I want to say about the integrated vector management. To key control dengue fever is adoption of a comprehensive approach through biological control, chemical control, environmental management, legislation, self-action and household and community steps. It is not only one step but a combination of many steps.
And developing control method for dengue are immature control and adult control as shown in this slide. We have also provided a Google survey form in which we have asked people how they can control, prevent, and treat dengue disease.
Here we can see that we are collecting mosquito larvae and then we will take it into our lab and put it in water filled container and do the experiment. Now we are collecting the water. Hello everyone, I am Tanvir from class 10 in Vidya Bharati school.
And I am Shobita Mishra from class 9 in Vidya Bharati school.
We are conducting an experiment in which we have collected samples of culex larvae and all other species of larvae as well from outside of the school, and now we are going to conduct an experiment in our laboratory.
Now, in our second step, we have taken a sugar solution in which we put the larvae that we collect in the morning and a simple water, tap water. We have put sugar in this solution so that larvae will get nutrition for its growth and changes into adult mosquito. This medium will be changed after a week.
Now, in the first step, I will put this tap water and sugar water in this container. Now, I will first make this dough wet and then I will put it over the container. I have put it over it so that the surroundings will become moisturous and it will be suitable for the growth of larvae to adult mosquitoes. Laboratory conditions for this setup are a temperature of 27 degrees Celsius and a management of 16 hours in the day and 8 hours in night and 1 hour in the break of dusk and dawn. We can also use another device which is sensors. Sensors are used to record sound of particular frequency produced by mosquitoes. Secondly, we can classify the type of insects by it. Here we can see that this picture consists of different versions of sensor. In the first figure we can see that this sensor consists of an observation pad, cup for carbon dioxide generating materials, and a camera and a lamp and sensor. And the second figure consists of heat pad, and the third figure consists of a sensor which is used to detect the mosquito, and then the drug package and dropper which is used to drop the drug which is used to kill the mosquito.
Thank you for watching our PPT.